The first topic in this unit that we are going to discuss is on biological databases. In the previous sessions, we have come through the concepts of data and databases and data mining, etc. And the applications of bioinformatics, its history, and some other general concepts. Now, our focus will be on more biological data-based applications and such databases and the data retrieval and similar other things. So that is the focus of this unit. Now, today I'll be handling biological databases. So let me share the presentation. Here you can see that biological databases are actually the storehouses of biological data. And why there is biological databases? Because there is a need for storing and communicating large data sets, and that need for storing and communicating large data sets has grown during these recent years because of the enormous growth of sequencing data and structural data and related other informations in biology, which is, that has accumulated in the biological, the different types of biological databases, demands storage and retrieval or data mining provisions for the scientists to access and generate useful information from that data. And of course, there should be large databases with search and retrieval provisions, and also that data needs to be made available to the scientists. So it's not only for storing and retrieval, that data needs to be provided to the scientists free of cost. So it's actually an information storehouse, and the public in general or the scientist who is working in that area needs to have access to the data. Then only our information will be available for further future R&D or research and development. So we need to store data, and there should be retrieval provisions or information retrieval provisions that it should be provided to the database, and also these data should be made available to the scientists for their research work, and also to make biological data available in a computer readable form. For example, previously about 20 years back, when I am doing my research for accessing biological data, I used to visit the Indian Institute of Science Library at Bangalore. And we have gone through the library where the hardbound volumes of different journals were kept, and we should take photocopies of the articles and take it back to the university and read it and take the extract as a handwritten notes. That's how previous, or at least just 20 years back, we are accessing biological data in such a hard copy format. But now, since the data in biology has grown enormously in such a logarithmic scale, and handling such huge volumes of data in manual methods of handling is quite impossible. So that there we require computer readable form of data and computer based retrieval and analysis of the data. That is almost required. That is very well required because of when we consider the volume of the data, because now we are handling big data even in biology. In biological database, the history of biological databases, as we discussed in one of the previous sessions of the history of bioinformatics, it's in 1965, Dayov et al. Published the Atlas of Protein Sequence and Structures. And I also mentioned that this is not actually a digital database or a computer database. It's only a book or a catalog type book in which the protein sequence and structures or the information related with all the available protein sequence and structures were published. And I told you a database can be a book or catalog. Everything can be considered as a form of data or a database. Okay. So this 65 version or 1965 version of this Atlas of Protein Sequence and Structures were not in digital format, but it's in a book, published book format. That can be considered as the first effort to catalog or to consolidate all the biological information related with the protein sequence and structures. So it is considered as the first biological database. Then in 1982, European Molecular Biology Laboratory, they initiates DNA sequence database, followed by within a year, just that is 1983, GenBank by the National Library of Medicine, NLM, of US. They created another database of GenBank, which is considered as the most popular primary database of nucleotide sequences. And initially, when it is created, it was established only in the LAN, local area network. It is not available at that time over the internet. Even the internet was made available and popular only in the 90s. So at the time, this library or the digital library or digital database that is created 1982 and 83 were basically working on the local area network of their own institute, and they created such a database based on in a computer readable form. And in 1984, just again 1 year after, Japan created another database known as the DNA database of Japan, which is also a primary sequence nucleotides database of Japan. And so within that period of 1982 to 83, 3 primary nucleotide databases to store the nucleotide sequences were created. And in 1988, these 3 databases, that is EMBL, GenBank, and DDBJ, that is a European Molecular Biology Laboratory and of Europe, and GenBank of US, and DDBJ of Japan, they agree on common grounds to share their data. So they and also to create a common format for data, because if you want to share the data and resources, usually it should be in a common, just like a common format. And you know that there are the cataloging of the libraries nowadays is universally accepting a common format, so that a book cataloged in India can be cataloged in everywhere elsewhere in the world, because they are following the same cataloging format for or classification of the books. So there is a standard format. Similarly, the data that is available in the biological science, especially in sequence, DNA sequencing and other protein sequencing information, that should be arranged in such a way that it should be acceptable and readable by the computers worldwide or the database worldwide. So they are actually adopted a common data format for sharing the data. And when we come to the public databases for molecular biology, we have these biological databases are actually libraries of life science information, as I mentioned, and they're collected from data of the biological database are collected from either from scientific experiments or published literature and computational analysis. Because, for example, if you are performing a sequencing experiment of your own, say, a 16S RNA, then that's the output of a scientific experiment. So that's one form of biological data. Or you are publishing an article in any research journal, that can be treated as a published literature, which is also another form of data. And if somebody analyzes already existing sequence in the database and creates a computer annotated protein sequence, then that's another data which is created by computer analysis. Okay, so there are different forms of data, like the output of the actual experiments, published literature, or even computer generated data, which is generated based on the other available data. Like as I mentioned, maybe a DNA sequence in database can be computationally translated into a protein sequence. So the protein sequence is not actually a sequence which is a sequence from the direct protein, but it is only a predicted output from the DNA sequence. That's a computation analysis result. Now, they contain information from research areas including genomics, proteomics, metabolomics, microarray gene expression, and phylogenetics. So different categories of information were stored in such biological databases. Information contained in biological database also include gene function, information, structural information like protein data bank where three-dimensional structures were stored, or clinical effects of mutations and similarities in biological sequences and structures. So there are various forms of biological data and specialized databases for handling such diverse information, or diverse types of information were nowadays available. So when we talk about the biological databases, we need to classify them. And basically, they are classified into two. That is a primary database and secondary databases. And there is another option where we have some aggregate of many databases, but that can it is not a different category. I will explain why. Anyway, we have primary database and secondary databases. Primary databases means they actually store the results of direct experimental output. For example, you perform your own laboratory experiment and you determine a structure of a structure of a protein or a protein. You sequenced a protein or sequenced a DNA fragment, and that is a direct experimental output of your research, and that can be given as an input to, for example, GenBank or protein data bank, wherever. And that type of databases where the actual experimental input from different sources were received, they were known as primary databases. In the picture, you can see that there are different sources. For example, scientist one who performed an experiment on DNA sequencing on a particular gene, and there is a scientist two who performed in his own lab with DNA sequencing on another protein or another gene. And similarly, there are different sources who actually conduct the experiments and generate the data. That will be directly given to the database. So this is a primary data which is actually obtained from experimental output, and that will be submitted by the original researcher. So these type of databases were known as primary databases. And the secondary database is another kind where the result of analysis of the primary databases were kept in another database. So in the next picture, you can see that these are the primary databases where direct input from the source is received. So a derived database or a secondary database receives the input from multiple databases, and they process the database, the data, and consolidate them into another value-added or more efficiently annotated database. So this type of databases, which actually depend on another database or different other databases to extract information and store and generate their own information, they are known as secondary databases. So they are the result of analysis of the primary databases. Now, aggregate of many database is also there. So it's not a different type of database like primary or secondary, but actually they provide link to other data items, or they provide a combination of data, or they provide a web search interface for data consolidation. For example, there are some search engines in which if you submit a query, it will search database 1, it will search database 2, and also it will search database 3. And it will extract simultaneously all the information from three databases, and it will be given as an input to you. For example, say there are a get entry or some other database. We will see in the next subsequent slides. Such type of databases, database search services, which access the data from primary databases and a single query. I will show you while we are performing the practicals. I can show you that. And for example, a single query on insulin in such a aggregate database search engine, it will access DNA sequence database of nucleotide sequence database, and it will list it will give a list of the entries of insulin which is present in the nucleotide sequence database. It will give you another list of entries that is given in protein sequence database, and also it can give you another list of entries which is present in the protein database where these three-dimensional structures are stored, and everything related with all the other different databases where such insulin related information is stored. Everything will be listed with their own total number of entries present. So you will get a list of different or you will get a list of all the databases where that search term is present. So such type of data aggregation or consolidation is possible by such aggregate services or aggregate of many database services. And we have some of the examples that we will discuss later. But basically, we have primary database and secondary databases, And the difference is that, okay, this is actually extracted from, I think, NCBI website. The primary database and secondary database were defined by them. And primary databases are populated with experimentally derived data, such as nucleotide sequence, protein sequence, or macromolecular structure, etc. Okay, the experimental results are submitted directly into the database by the researchers, and the data are essentially archival in nature. So that type, the data is actually intended for storage purpose. So that is a primary database. Whereas the secondary database is it comprises of data derived from the results of analysis of the primary data. They are often referred to as curated databases, but it is not exactly true, because you can say that it's a misnomer, because primary databases also are well curated nowadays. For example, if you want to submit a DNA sequence into GenBank, which is a primary database, they will ask for all the related informations. What is the nature of the gene, from where did you, or which is the organism from which you isolated, what is the sequencing method, and all other information, even the other information will be added to the data. Simply, instead of storing that information or the sequence information directly in the database, all other related information, available information, will be attached to that data. So that is actually the process of curation. So without any label or without any identification mark, the data doesn't mean anything. From where it is isolated, or what is the nature of the data, it needs to be described with that data. And that means most nowadays primary databases are also curated, but the degree of curation or degree of additional data that is added or description that is added, that will be higher in case of secondary databases, where usually manual verification of description of the data is done. And it is not only manual verification, there are computational algorithms. For example, they are highly curated. In the case of secondary databases, they are highly curated, often using complex combination of computational algorithms and manual analysis and interpretation. So using computational algorithms and also by manual verification, the data in the secondary database were more curated. Curated means they are well described by adding additional information or all other related information with that, or the different descriptions about the data that will be provided in the secondary databases. So this is the difference between primary and secondary database. And here's a comparison table in which the primary database is actually an archival database, which is mainly intended for storage purpose, whereas the secondary database is a curated database with well annotations or annotations or descriptions added, and it can be considered as a knowledge base. Then source of the data is actually direct submission of the experimentally derived data in case of primary, whereas results of analysis, literature search, and interpretation of or further the data generated from the primary databases were stored in the secondary database. Then there are different examples like DDBJ, GenBank of nucleotide sequences, and Array Express or Archive or Geo, that's functional genomics data, is stored in such type of databases. Then three-dimensional coordinates
Excuse me, sir.
Yes. Yes.
Sir, screen clear ആവുന്നില്ല. Presentation is not clear.
Presentation clear ആവുന്നില്ല. Maybe there's a problem with the bandwidth, because it's a HD broadcast, that's why.
Okay. So will we get this from elsewhere?
The entire HD resolution video will be provided in the platform. Later you can
Moodle.
Moodle
Okay.
platform. You will be getting that one. So that will be
Thank
clear.
you, sir.
So right now you can listen to me,
Yes,
and
sir.
if the text is not visible,
Okay.
you will get another copy with a very clear text, HD resolution. Right now,
Okay.
if you have some
Okay, sir.
limited bandwidth, certainly that limitation will be there, because it will not be clear because of the limitation in your bandwidth.
Yes, sir.
Okay.
Okay.
Okay, fine. So what I'm talking is these three-dimensional or macromolecular structures that will be stored in a primary database like Protein Data Bank, and there are Interpro and UniProt or Ensembl, etc. There are such knowledge bases where secondary or derived data is stored. Some of the examples are there, and we will see some of them in our later sessions. So this is the general concept of about the primary and secondary database. Now, when we come to some of the examples of biomolecular databases, based on their different functions. They can be categorized. Sequence and structure-based database are there, which store primary sequence information like DNA sequence or protein sequence or protein structure, and that they are structural databases. So sequence and structural prominent sequence and structural databases are UniProt, for example, Universal Protein Resource, that is UniProt. That is for a primary protein sequence. And then DNA sequences, we have three primary databases. That is a European Molecular Biology Laboratory, that is EMBL, GenBank of US, and DDBJ or DNA Data Bank of Japan. There are the three primary nucleotide databases for DNA sequences. For three-dimensional structures of the protein, where we have three-dimensional visualization files that is available, and that is a PDB or Protein Data Bank. Then structural motifs of the proteins, that's another catch, that is another database which is stored with the structural motifs of the protein. Sequence motifs are actually stored in prosites and prodome, etc. So these are protein related or protein structure related terms, the motifs and other things. So structure and sequence motifs will be that information, related information, and their annotations and descriptions will be provided in such a specialized database like a prosite or prodome, etc. Now, genome sequence and an entire genome, not only a single fragment of the sequence like a partial sequence of a cDNA or a protein. The entire genome sequences and their descriptions, their annotations were provided in genome-specific databases. Some genome-specific databases are like SGD, that is a Saccharomyces genome database for storing the entire genomic data of Saccharomyces. Flybase is for another one, and then ESDB, PlasmoDB, etc. There are specialized databases for storing entire genome sequence of a single organism. So, specific organism specific complete genomes were stored in such databases. Now, multiple genomes were actually present in INDER8 or NCBI, KEGG, TIGR. So these are some of the examples of databases in which multiple genomes or genome of many organisms were stored. So genome specific or species specific databases are there, and even some databases were there which contain genome sequences of all the organisms. Okay, complete genome sequence of all organisms. And then based on molecular functions, we have some database like transcriptional regulation, enzymatic catalysis, and transport, etc. Then biological process database are there, metabolic pathways, for example. KEGG is one of the internationally known or well-known database, and KEGG is actually, I think it's from Japan, KEGG. Kyoto Encyclopedia of Genes and Genomes, that is KEGG, which is a very interesting database in which all the metabolic pathways were well described. So if you have some interesting enzyme, for example, you give the name of the enzyme in the metabolic pathway search interface of KEGG, and all the pathways which is related with the other interacting components will be shown as a flowchart or like a metabolic pathway chart. That will be, and other annotations and descriptions will be available in the KEGG interface. So biological processes and their interactions can be easily accessed by such databases. Then signal transduction pathways, protein-protein interaction, and gene networks. So there are a lot of databases, maybe thousands of databases are there for individual specialized functions. So basically, we have sequence and structure databases, then entire genome collections, then database based on molecular functions, and that then another one, databases based on biological process informations. So these are some of the categories. It's not complete, but it's only a few of the most popular categories. We have thousands of such specialized databases available nowadays, which store DNA sequence information, structural information, or molecular functions, or protein sequence motifs, and even biological process related information, etc. Is it clear? Hello?
Sir.
Is it clear?
Yes, sir.
Okay, fine. So now, this is the international collaboration of the three primary nucleotide sequence databases, as I mentioned. NCBI, that's from US, National Center for Biotechnology Information, and that DDBJ, that's DNA Data Bank of Japan, and European Molecular Biology Laboratory, EMBL. So these three are the NCBI database is GenBank, then EMBL and DDBJ. These three are the three primary nucleotide sequence databases which collaborate from 1988 onwards. They are actually collaborating to share their data. So, which means we have a common sharing system. If you submit, for example, you update or submit something in the NCBI database, you are generating a new DNA sequence, or you are sequencing your own 16S RNA, and you are trying to submit it into NCBI. Once it is submitted to NCBI, it will be within a minimum period of time, it will be shared with the other two. So it will be synced. That syncing process will result in synchronization of the data of the three databases. So it will be made available to the one who is using these CIV, or say what is called Japanese interface, or European interface, or European database. So in all the interfaces who are searching with their query, they will be provided with the same information that is submitted in the GenBank. So that means, so there is a sharing system. The submissions and updates which is submitted to one database will be shared or synced with the other databases, and that will be made available to the others. So in this system, this is the National Institute of Health that is actually holding the NCBI database. Then this is the DDBJ search interface and their institute. This is the European Bioinformatics Institute that is holding the EMBL database. Now they have a GetEntry. GetEntry is the search system that is provided for getting information from DDBJ. Endress is the search system that is provided by that database for searching their own database. SRS, or sequence retrieval system, is provided by EMBL for searching the database. So this is actually a web interface, just like a Google search engine, or they provide their own search interface. Once you visit the website and search for something, they are actually executing this meta search provision. And this is actually an aggregate database service. Aggregate means the NCBI, because NCBI GenBank, they contain not only the DNA sequence, but they have protein sequence and other related information. Maybe hundreds of different other bioinformatics related databases were there in GenBank. So a single query that is submitted to the Endress search engine will search across all the databases provided in GenBank. All the databases means it may have DNA sequence, protein sequence, express sequence tags. Then similarly, we will see the list of them, the other databases when you visit the site. So all the different databases which is present in GenBank will be simultaneously searched with a query that is submitted. For example, you are submitting an insulin search, the term insulin into the search box of Endress search engine, that will search across all the available databases in GenBank and related NCBI databases, and you will get a result. And that means this is a meta search provision or an aggregation, just like I described in the classification of the databases like primary and secondary, and also there are aggregation services which will search across other databases. So these SRS, GetEntry, and Endress, they are such a aggregation database aggregation services for information retrieval. Now you come to the growth of GenBank and other databases. This is an old picture. We don't have a recent one, but you can see from 1998 onwards, the exponential increase in the growth of total number of base pairs in the database. So during this small period of these 15 years or something, oh sorry, it's about 20 or 17 years, right? 20, more than 20 is there. 80 to 92, 27 years. So during that period, but the exponential increase starts from 2002 only. By the period only very few bases were added in the database. But since the beginning of the big data generation, that is from 2002 onwards, you can see an exponential increase. I mean, within about 5 or 6 years, we have very significant growth in the number of base pairs. In the total number of sequence related information that is submitted in the GenBank. But this is another one, another graph that shows 82 to 2019 of GenBank that shows this is the growth, and this is all genome sequence. This red line is all genome sequence, and this is the gene sequence, so GenBank. So the complete sequences or sequence related information or the total number of bases that is submitted in the GenBank database again increases exponentially. So this is a basis, and this is also another similar type of sequences. It's a graph taken from the NCBI site, and this statistic shows you the increase or exponential increase of the size of the database during this period. This is a EMBL growth. There also we can see that there is a exponential increase in the number of bases. For example, here in this case, about by 2012, they have about 411.9 billion base pairs sequences that is submitted in the GenBank. So, as I mentioned, the volume of the data that is stored in such database is very huge. And this is about the challenges for bioinformatics. Since there is an exponential increase in the growth of the sequence information. So here Human Genome Project is there, then by 2000, and again this increase, this is only up to 2000. From 2002 onwards, it is exponentially further increasing in the same pattern. Okay, so after 2000, we have the generation of the big data by parallel sequencing instruments and other things. So the whereas the protein structure prediction and function prediction and other related published literature based on this. So the publications actually which uses this information were very low. So we have a very large gap, and this gap means this sequence information is not yet explored. For example, there may be unknown genes or functional genes that can be identified, and their function can be predicted, and they can be expressed in some condition. So for identification of an unknown or is not of a gene which is not yet expressed and identified, you can analyze the entire DNA sequence information and locate the possible genes and see the protein or predict the protein, how it is translated, and what will be the structure and function of the protein, etc. can be identified. So possibly you can explore the entire genome information for generating further possible useful outcomes like identification of novel genes or gene products, etc. But that type of research is required because our actual wet lab experience, which is depending on such a laboratory work, that is a protein structure determination or sequence determination or its biological function or activity determination in our own laboratory, that phase of growth will be very slow. But if you can use such computational data to predict the possible genes, their functions and other things, then the blind exploration or random exploration of possible protein producers or protein targets, etc. can be minimized. So we have a huge amount of data available in the database that can be properly processed to generate or extract more information, and that is the challenge for the bioinformatician, to analyze the data and extract useful information and predict the possible genes, their functions, their interactions with the biological systems, etc. Okay, now we come to the NCBI homepage. NCBI is National Center for Biotechnology Information of US. It's a part of the National Library of Medicine. And that National Library of Medicine actually they holds these database of GenBank. GenBank is actually primary nucleotide sequence database. Other than that, they have a lot of other databases and other services. So in the home page you can see, and this page is actually an old image, maybe 2 or 3 years back I taken this screenshot. Usually this look and feel of such websites will be different from what I am showing in the slide, because they will periodically they will revise the look, and the provisions will be different. So maybe the layout of the web page will be different if you are visiting right now. So anyway, the basic concept is that all such websites, all such database services, whether it is NCBI or Protein Data Bank or Swissport, they all provide a search box for giving your input in the first page itself, in the home page itself. And they also, if they have multiple databases, they will also provide a pull-down menu for selecting the desired database if you want a specific database. For example, if you are looking for express sequence tag, you can select express sequence tag EST database. Or if you are looking for a protein sequence of insulin, then you can select protein database from that pull-down menu and give insulin in the search box. Similarly, that search box can be used for giving the input, and also you can specify the database into which you want to search. Because I already mentioned that these NCBI databases where it's an aggregate, and they have a lot of other related databases other than GenBank, which holds the primary nucleotide sequence information. So all such websites, all such database websites will provide you with a search service or a query service into which you can give the input to get the exact information, or to extract the information from that database. So I selected these all database pull down menu. I have selected these as nucleotide sequence. Nucleotide sequence means it will search in the GenBank database, and I have given the query term insulin and submitted the search. So it returned, say, maybe there are many pages are there, and out of say 1,81,535, that much entries are there, insulin related. So the entry means that this means somewhere in the title or description the term insulin is there. So that's what it means. So somewhere in the title or in the description, the term insulin is used. That's why it is listed. So all the listings, all the subsequent submissions in the database, GenBank database, with insulin in the title will be displayed. So that may be insulin or insulin-like, insulin receptor, insulin precursor, insulin binding protein, everything will be listed. It's not only insulin. So you have if you want to recover the sequence of the insulin, you have to look in the number of base pairs and other descriptions, etc. Because the entire chromosome that contain insulin may also be listed. So here you can see that there is a linear DNA sequence about more than 3 lakh base pairs as a trade entry, which is not actually insulin gene. So a complete cDNA sequence is listed, and insulin precursor is listed. So the concept of this query is that everything in the title with the term insulin will be listed. So it's not the exact insulin gene. So you can filter the results using different criteria, like for example, Homo sapiens. You want on the right side there will be a Homo sapiens. In brackets there is a say the number of entries there. So if you select one of the organisms, the results will be filtered only, or it will be limited only to the concerned organism. Or if you are looking for some bacterial or other things, then you can select that one. Only that particular category will be selected. So you can apply different filters and other things during the search. And anyway, that will be discussed in the practical sessions. So, and based on which you can filter the results and get your concerned sequence or retrieve the concerned sequence, and that's how it will work. So this is one of the examples of the data, GenBank entry data. And in the last class, I mentioned that there is a flat file, or a flat file format for storing such information. So this is a GenBank flat file. If you open one of the entries in GenBank as listed here, if you open one of the entries, it will look something like this. So there is different descriptions like locus, definition, accession, version, keywords, then references, or the title. So there are a lot of descriptions, and the data is provided here as a text file, just a continuously written text file. So this is how, and at the end there will be the sequences present. So all the descriptions, after all the descriptions which is written in a flat text file under different headings, there will be the actual sequence of that particular gene, which is given in the single letter code. Now, let's see what are the different regions in that flat file means. So here you can see that the first one, locus, location of that particular gene. So this is the locus name. This is the sequence, actual length of the sequence that is attached. And this is the type of the sequence. Actually, it's an mRNA sequence. Then other database version and submission date or modification date. Submission or modification date is provided. Then definition means from where it's a definition of, like, actually it's a title which describes the nature of the sequence. Okay. This one is the accession number, just like any catalog entry in a library. It requires a unique identification number or accession number, and the version is it's a 0.2 or 0.3. That's the revision version number. If somebody else revises that sequence after further research, then there will be another version number which will be appended at the end. 0.2, 0.3 will be added. So this is GenBank identification number or GI number. So all these are part of the flat file description. Then keywords. If something is provided as keyword, the search results will be looking for the keywords in this entry. And for example, insulin or insulin gene, etc., whatever you can type there. And that keywords will be keywords is what the search engine looking for. And if you provide such a specific keyword, certainly that if somebody else search with that keyword, this entry will be listed in the search results. Then this is a source from which it is isolated, and this is a scientific name of the organism from which it is isolated. This is a common name and a scientific name. Then finally, there is other descriptions like the references and means the article in which it is published, and the author who published it. All such informations will be there. And finally, there will at the end there will be the actually coding sequences which will be provided at the end of the flat file. So this is how an entry in GenBank or even in other databases, they actually follow the same format, a flat file entry. It will have all these provisions. So if you read these flat file, you can understand all other related information about the sequence. So it's not, as I mentioned, it's not a simple sequence that is submitted, but it is attached with all other descriptions and related information, and that can be retrieved from the database. So the data or the sequence data in such a primary database like GenBank were stored as flat files with all the related descriptions or annotations, or actually that's why it is called curated, and they are stored with all other related informations or annotations. So this is the record, or the actual sequence is present at the end, and it is closed with a double slash, or that symbol is the end of the record. So that means that that's the end of the concerned flat file record. So this is how a flat file entry is kept in the GenBank, which contain all the sequence and related informations. So this is another one from PubMed, another one from same NCBI. The GenBank is their primary nucleotide sequence database, whereas the PubMed is the literature database of GenBank. If you want some specific scientific literature, PubMed is your best choice. You can, for example, SARS coronavirus or COVID-19, you select the search term, or the database may be searched as selected as PubMed instead of nucleotide. If you select nucleotide, it will look for DNA sequence or RNA sequence. If you select PubMed, it will look for the concerned query. For example, SARS coronavirus or COVID-19, whatever search term that you are giving, it will look for the literature information or published papers, scientific papers related with that term. So PubMed is the literature database, and this also an old slide, and at that time, at least 2 or 3 years back, there were 19 million citations, but now again you can see that it will be 23 or 25 million citations, at least it should be there. Now there are other databases, as I mentioned, the NCBI, other than the nucleotide sequence database or GenBank sequence or protein sequence database, and these PubMed, which is store the literature information, there are other databases which is present in NCBI. EST base is one of the example. It is expressive sequence tags or the cDNA, single pass reads of cDNA information is present in the EST database. Then another one is genome survey sequence, that is GSS database, hologene, HTG sequence, single nucleotide polymorphism database, a reference sequence database. Means it actually contains reference sequences, because there may be the same insulin gene can be submitted by thousands of people. So there should be a reference sequence to find to verify which sequence is actually mutated and which direction. So that such type of reference sequences database is also present in NCBI. Then Unigene. So these are all different specialized databases provided in the NCBI, other than the GenBank and the PubMed. So now this is about EMBL EBI. Anyway, the basic concept is same. If you take the case of GenBank or EMBL or DDBJ, all contains the same data, or because they are sharing the data, the same flat files or same sequence information is present in all the databases. But the front end means the search interface and the front end, that will be different. But still they provide with their own database search option in the home page, and the query results will be different, because that depends on the concerned institution that is maintaining that website. So their algorithm of extracting the information is same, or the data is same, but the algorithm that is used to analyze that data and generate useful information will be different in 3 interfaces. So most probably or widely you can say that GenBank is popularly used, and because their extraction of information and presentation of related information will be better, if at least I feel so, because compared to the EMBL and DDBJ, it's most popularly used, and their data extraction and the way of presentation in the results will be much more better than the EMBL and DDBJ. Even though they contain the same information, how it is presented to you after the extraction will be different. Okay, they will provide the search box, but still many of people prefer the data extraction methods, or the say endless search engine and data presentation or extraction presentation in the GenBank more useful. Okay, so this is the overall size of the say EMBL database and up to 2019, and this is another one, that is a DDBJ. Their homepage is shown, and they also provide with multiple databases of different categories, and a search engine is also provided. You can search for the sequence in that one also, and there are provisions in the database and other value-added service in the database will be different than the GenBank. So these are all some of the metadatabases. Metadatabases means like a collection of data. The third category, as I mentioned, they are usually aggregation services, or which extracts the information or collect data and present to you from various databases. This can be considered as a database of databases. They collect data from different sources and usually make them available in new and more convenient form, or with an emphasis on a particular disease or organism. So we have NRES of National or NCBI, National Center for Biotechnology Information, and UGenes or gene cards or source and bioinformatic harvester. These are some of the examples of such metadatabases which extract information or consolidate data from multiple databases. So they are known as meta databases. So this is another one, that is Ensembl. Ensembl genome browser is shown here. Ensembl is another database. Ensembl actually they contain the entire genome information, graphically presented genome information. So for example, if you take the case of all genome of humans, we have 23 pair of chromosomes. Each pair of chromosome is actually listed graphically there, and you can graphically browse the genome. And usually in other databases, you can give a query and the flat file will be listed there. But Ensembl browser allows the access of the entire genome of the organism based on their chromosome. And for example, if you select the chromosome 1, then graphically you can select the chromosome 1, and that will be opened. And at the top page, you can have the chromosome 1 represented here. If you select an area by placing the cursor, just like this one, the red line that is shown here. You place the cursor there, then then the subsequent windows or the bottom portion shows an expanded version of the different locations or loci of the gene in the expanded window. Again, if you select one of the areas that concern the gene, will be displayed on the bottom portion. If you select that basis or a period of the basis, then that will be that area can be selected, extracted area, or the sequence in can be extracted from that database by graphical selection methods. For example, if you want to download the sequence from 25,000 to 23,000 or 25,000 to 28,000, then you can select that area of the sequence from the graphical browser, and you can download that sequence of the chromosome based on the or you can, if you select one entire gene from this location, you can completely select that entire gene and download the concerned sequence of the gene by browsing it graphically. So that's one of the features that is available in Ensembl. So this is about primary sequence databases of nucleotides and some general concepts. And the next topic is proteomics tools that we can discuss in the next session. Is there any questions?
No, sir.
Okay. Fine. Thank you.